Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base biotech podcast, and this is number 71, and it's going live on August the 14th. I think we're in heat wave number 4 now in the UK for the summer, and it's even warm up here in Scotland. But it is cloudy, and so that meant no clear skies here for the eclipse this past week. Watching it on the news really isn't the same thing. We also were filming in St Andrews last week. That's not the one in Canada, it's the one in Scotland, and I was attacked twice, once by a very angry wasp, and the other was a very hungry herring gull that flew into my head to try and knock a sandwich out of my hand as I took a bite. Clearly it didn't know me and my love of food, because I held on to the sandwich even though it was like being punched. There are signs everywhere in the town saying don't feed the gulls. Well, I managed not to, just. I wasn't really angry, they're just hungry. It always amazes me how people get mad at animals going after food or coming into urban areas. We're the ones that destroyed their habitat, not the other way on. What are they supposed to do? Anyway, moving along. Last time we had a conversation about GI motility disorders with Peter Milner, director and co-founder of Renexion. This week we have a conversation with Paweł Szczepański, CEO of JJP Biologics, about specially built antibodies to tackle autoimmune diseases and cancer. If you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com, which is also the address you can use if you're interested in advertising with us or sponsoring a podcast episode or for event coverage. Normally at this point we'd run through some of the news headlines from articles you may have missed over at deeptechdigest.com, but I think everybody's been on holiday, so there wasn't a lot to choose from. There's definitely news worth reading though, so do head over to deeptechdigest.com and check them out. And so this week we're off to Poland, although not literally, much as I would like to. This week we have a conversation with Paweł Szczepański, CEO of JJP Biologics, about specially built antibodies to tackle autoimmune diseases and cancer. So hi Paweł, thanks so much for joining us this week, and I'm looking forward to hearing more about what you do. Could you start with a bit of background on the company?
Sure. So JJP Biologics is a clinical stage biotech company. We're based in Warsaw, Poland. What we do is actually develop precision antibody therapies for autoimmune disease and cancer. And our particular interest is in immune pathways that have strong biological relevance, but have not yet been fully translated into medicines. So our starting point is always the disease mechanism. We ask, in principle, at the beginning of the process, 3 main questions. Which immune interaction is driving the tissue damage? Whether it can be addressed selectively, and I will get to that later on, and whether there is a credible path to demonstrating target engagement and patient benefit. And only from there, when we have 3 positive answers to those questions, we design an antibody program around that biological hypothesis. And this matters, because many patients still do not achieve durable control with the existing therapies. We all know that. Some treatments broadly suppress the immune system, some work only in a proportion of patients, and other therapies control symptoms, but without sufficiently changing the course of the disease. Our ambition at JJP Biologics is to intervene more precisely into the pathway responsible for the disease itself. How we do it, we combine the focus and the creativity, if you wish, of a biotech company with the long-term backing from the Starek family and the operational experience of Polpharma Group. And those 3 ingredients give us the stability to pursue differentiated mechanisms and build programs from discovery through clinical development.
How does that differ from what other companies are doing?
Well, I do believe that we do things differently. Our approach is, we put it biology first and mechanism led. So rather than beginning with a fashionable target and then looking for indications, we begin with a disease in which the underlying immunology points to a specific pathogenic reaction. As a scientific team, we are particularly interested in functional selectivity. So the goal is not simply to bind an immune receptor, but to block the harmful signal while preserving as much useful immune function as possible. And that is an important distinction. In both autoimmune diseases and oncology, where we all know that excessive suppression or excessive activation can create problems. So what we are doing, we are especially interested in immune pathways that may have been underestimated by the broader industry. And a good example of this is the IgA CD89 pathway. For years, the field has concentrated heavily on IgG-mediated biology, B-cell depletion or broad cytokine inhibition. We saw a compelling evidence that IgA-driven inflammation could represent a major therapeutic opportunity, and we built an entire platform around that insight. So the difference is not novelty for its own sake. It is selecting a less explored mechanism because the disease biology supports it, and only then developing the molecule, biomarkers, and clinical strategy around a testable scientific hypothesis. So ultimately, our goal is not to compete by being incrementally better. Our goal is to open entirely new therapeutic avenues for patients.
Obviously, you're in Poland. I wonder if you could tell me a little bit about the biotech and drug discovery scene in Poland and whether it's concentrated in Warsaw or whether there are other hubs.
Well, I must say that I was expecting that question, and it's quite clear that Poland is maybe not the top geographies that come to your mind when thinking about innovation in biotech. But Poland's life science sector is changing quickly. And what some of us know is that the country has a strong foundation in pharmaceutical manufacturing, especially generics, but also clinical research, medical science. And that base currently is increasingly supporting innovative biologics and original drug discovery companies. And in Poland, Warsaw is an important hub because it brings together universities, research institutes, specialist hospitals, so important for early stage clinical trials, but also experienced clinicians, investors, but also a growing number of biotechnology companies. So we are very proud to build JJP Biologics here while still operating with global development mindset. At the same time, the Polish ecosystem is broader than Warsaw. There are academic and biotechnology communities in several Polish cities, each with different strengths. So JJP Biologics, for example, is building an R&D lab currently in northern Poland, in the beautiful city of Gdańsk, exactly to be able to benefit from the local talent pool there. So yeah, Poland has an excellent scientific and clinical talent. To turn that into a globally competitive biotech, we need patient capital, experienced development leadership, strong international networks, and simply confidence to build valuable intellectual property here in Poland rather than exporting the ideas prematurely.
I guess back to the company now, but what conditions are you working on?
Our lead work is in autoimmune and inflammatory diseases in which IgA biology may play an important role. These include linear IgA disease, rheumatoid arthritis, IgA nephropathy, IgA vasculitis, and lupus-related renal disease, which I admit seems a long list for an early-stage asset. But what is really exciting is that although this disease appear very different clinically, there is increasing evidence that they share common biological drivers involving IgA-mediated immune activation. So IgA can engage CD89 on myeloid immune cells, including neutrophils or monocytes. When that signaling becomes dysregulated, it can amplify inflammation and contribute to tissue injury. So looking from the other side, that opens the possibility of creating a platform approach where a single mechanism can potentially address multiple diseases. Obviously, remain disciplined. So each biological rationale must be tested separately in each disease with the right patient population, endpoints, and biomarkers. And in parallel, it's probably worth also mentioning that we are developing an oncology platform and programs focused on, once again, underexplored mechanisms that regulate anti-tumor immunity and the tumor microenvironment.
So could you tell me a bit about how your antibody engineering process works?
Well, what makes JJP Biologics stand out is the fact that the entire early stage discovery process is carried out by our in-house team. So on our IP documents, you will see names of our Chief Scientific Officer, Louis Boone, and other discovery team members. Of course, this does not preclude bringing in from the external world great science in collaborations in the future. So without discussing proprietary details, the process begins with target biology and the desired function. What we do is we design the disease-driving interaction, so to say, the interaction that we want to interrupt, then generate and select antibodies based not only on binding, but also whether they produce the intended biological effect. And the next stage is optimization. We look at specificity, potency, stability, manufacturability, a key word recently in the industry, and the characteristics needed for a suitable therapeutic profile. So an antibody must be biologically compelling, but it also must be robust enough to manufacture consistently and develop as a medicine. Then we connect laboratory findings with translational evidence. That means identifying pharmacodynamic measures or biomarkers that can show whether the antibody is reaching and engaging the target in humans. So this link between mechanism, molecule, and measurable clinical biology is what's central to our approach. And the aim is to enter clinical development with a clear chain of reasoning. This pathway contributes to the disease. This antibody modulates the pathway in the intended way. And these clinical measurements tell us whether the hypothesis is really working. And what I find most rewarding is when all of those layers come together and the clinical data begin to confirm the biological hypothesis. That is where science translates into potential benefits for patients.
And so could you tell me a bit about the IgA CD89 axis and the relevance of that in disease?
Sure. IgA is one of the major antibody classes in the human body and is especially important at mucosal surfaces. Under normal conditions, it contributes to immune protection. But in certain autoimmune and inflammatory diseases, IgA autoantibodies or IgA-containing immune complexes can become part of the disease process. So CD89, also known as the Fc alpha receptor, is found mainly on myeloid immune cells such as neutrophils, monocytes, and macrophages. When pathogenic IgA immune complexes engage and cluster CD89, they can trigger cellular activation and inflammatory responses. So depending on the disease, this may contribute to tissue injury in the skin, joints, kidneys, or other organs. And from literature searches, it was revealed that in many autoimmune disease, patients that have disease-specific IgA autoantibodies have more severe disease and respond worse to current available treatments. So the conclusion was quite clear to us. The therapeutic idea is to interrupt the harmful interaction between IgA and CD89 without broadly shutting down the immune system. This pathway has received much less therapeutic attention than several IgG-related mechanisms, despite substantial body of biological evidence linking IgA to the disease itself. So for JJP Biologics, This axis is important because it potentially connects several indications through one mechanism. Our clinical work is therefore designed to determine whether blocking that mechanism can translate into meaningful outcomes for patients.
And so what is JJP? Is it 1212 or 1212? I guess it's the same thing, but
Yeah, so our internal code name is JJP1212, but it's also known as nebaprubart, and that's our lead clinical candidate. It's a first-in-class humanized anti-CD89 antibody designed to act as an antagonist of the IgA CD89 pathway. In practical terms, nebaprubart is intended to prevent pathogenic IgA from activating CD89 positive immune cells. And by interrupting that interaction, the aim is to reduce the downstream inflammatory cascade while avoiding broad immune cell depletion. That's the safety aspect to it. Rather than broadly suppressing immunity, we just simply target a specific pathogenic pathway. So we are evaluating the molecule across several IgA-driven diseases, including rheumatoid arthritis, lupus, and IgA nephropathy. But the program has also received an orphan medical product designation from the European Medical Agency in linear IgA disease. So that reflects in our understanding both the unmet patient need, but also the novelty of the approach. And if successful, nebaprubart, or JJP1212, could establish the first clinically validated therapy targeting CD89-mediated diseases.
so could you tell me about the clinical trials that you have in place and where each of those is currently at?
JJP1212 has completed the initial clinical evaluation in healthy volunteers, where the program assesses safety, pharmacokinetics, immunogenicity, and pharmacodynamic evidence of target engagement. And that work provided the foundation for studying the molecule in patients. Currently, our phase 1b study in linear IgA disease is ongoing, and we announced positive interim data in June 2026. It's an open label study. The significance for us is not only the clinical observation, but the opportunity to test whether the mechanism we identified in the lab is translating into patients. We are also advancing a phase 1b program in rheumatoid arthritis with patients dosing expected later this year. And this will allow us to evaluate the same pathway, but in a larger and systemic inflammatory indication with a different clinical presentation. And in oncology, our second asset, JJP1008, is progressing towards a phase 1/2 clinical evaluation with trial approval anticipated also later this year. Across the portfolio, our priority is discipline and clinical execution and generation of interpretable data that can guide the next development decisions. That's our short-term goal.
And at the beginning, you were mentioning about treating people with disease. What are your hopes for patients in these areas? Is it just making disease manageable or curing disease? How do you see that?
That's the perspective. Thank you for that question, because that's the perspective that really is close to my heart. So my hope is that we can move beyond symptom management and really begin addressing the biology that actually causes the disease. In many autoimmune diseases, treatment still depends heavily on broad immunosuppression or on cycling through therapies until something works. And that, we all know, can leave patients with incomplete disease control, cumulative toxicity, and considerable uncertainty. So with JJP1212, the biological ambition is to interrupt disease-driving IgE signaling rather than suppressing the immune system as a whole. If the mechanism is validated clinically, that could provide a more targeted option for patients whose disease is driven by this pathway. And according to our conservative data, there are more than 2.5 million patients awaiting such a therapeutic option, only in the indications I have mentioned earlier. So the long-term goal is disease modification, deeper control, and where possible, durable remission. Of course, we should be careful not to promise outcomes before they are demonstrated, But that is the standard we are working toward. Not simply making symptoms temporarily better, but changing the biology that sustains the disease. And we have data to support that claim. We also see the therapy as potentially complementary to the current standard of care. Precision medicine often advances by adding the right mechanism for the right patient rather than assuming one treatment will replace everything else.
And so could you tell me a bit more about JJP 1008?
Yeah, that's the second program I mentioned in oncology. So it's actually our lead oncology program and targets HVEM. HVEM is also known as CD270, which is a context-dependent co-signaling receptor. BTLA and CD160 bind its CRD1 region and can deliver inhibitory signals, whereas light engages other extracellular domains and can support immune activation and survival. So in cancer, tumors can exploit inhibitory immune signals to reduce the activity of T cells, while JJP 1008 is designed to interfere with inhibitory signaling associated with the HVEM pathway and thereby supports restoration of anti-tumor immune activity. What interests us scientifically the most is that the fact that the strong expression of HVEM strongly correlates with, unfortunately, quick progression of the tumor. So HVEM represents a different checkpoint biology from the pathways addressed by the currently established checkpoint inhibitors. And once again, for us, that creates the possibility of showing activity in settings where existing immunotherapies are insufficient and the potentially of rational combinations with established agents.
And was that one on its pathway through clinical trials?
Yes, so JJP 1008 is moving through the final stages, I would say, of preparation for first-in-human clinical development. The program has undergone the non-clinical studies required to support the clinical application, including the pharmacology and safety assessments. So the planned phase 1/2 study is intended to begin with a careful evaluation of safety and dose, while also exploring pharmacodynamic and early anti-tumor signals in patients with advanced solid tumors. And that's to happen early next year. Because this is a novel checkpoint mechanism, the early clinical study must be informative rather than simply procedural. So we want to understand exposure, we want to understand target-related biology, patient selection, and the potential role of combinations. And those findings will shape the most appropriate development path looking forward. Trial approval is anticipated later this year.
You're already doing a lot, but is there anything else in your pipeline other than that?
I might surprise you, but yes, of course. The most immediate opportunity, thinking about the assets I mentioned, is to expand the clinical understanding of JJP 1212 across additional indications, mainly the IgA-driven disease. So IgA nephropathy, lupus nephritis, and IgA vasculitis are for us particularly important areas because the disease burden can be substantial and the underlying biology offers a very clear rationale for investigating the CD89 pathway. So we're also working on initiating a trial in the renal indications for our lead asset. But we are also developing a third asset, JJP 2409, which is an oncology program focused on an angio checkpoint concept with the tumor microenvironment. So at this stage, the most important fact is that it follows the same company strategy. Identify a biologically important but insufficiently addressed mechanism and build differentiated antibody program around it. But beyond individual assets, we are building something more, an integrated capability in antibody development, and that includes target assessment, antibody generation, translational biology, non-clinical development, manufacturing strategy, and last but not least, clinical execution. So as you can clearly read or hear in between the lines, the broader ambition is to build a sustainable biotech company from Poland, but with global scientific and commercial relevance. For us, success means creating medicines that can change outcomes from patients, demonstrating that innovative drug development can be built and led from this part of Europe. So we are building JJP Biologics in Poland with global ambitions, but our purpose is very straightforward. Understand disease biology deeply, translate that understanding into precise medicines, and create meaningful new options for patients. If we can combine scientific courage with disciplined clinical development, we believe that we can make a real difference.
Is there a geography associated with this? I mean, are you concentrating on Europe first, or are you trying to do this globally?
The clinical phase 1Bs to As that I mentioned will be executed predominantly in Europe. But of course, the therapeutic potential of the therapies is global. And that's how we plan to expand it, either independently or through partnerships with partners who see the similar potential in the assets as we do.
Well, it was nice to learn more about some of the groundbreaking things JJPBio is doing, and also a bit about biotech in Poland as well. I always enjoy going there. Some great restaurants in Warsaw. I think that's definitely a worrying pattern there, where I describe cities by their food. So that's it for this week's podcast. If you do want to get in touch with guest opportunities, or if you want to advertise, please do feel free to contact us at jim@deeptechdigest.com. And so until next time, wherever in the world you are, I hope you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.